today is like we are facing a lot of bottlenecks in analyzing the data when we deal with the organism whose genome or the transcriptome has been not sequenced yet. So I hope the screen is visible to all the slides.
Yes, sir.
Yes, sir.
Yes. Okay. Yeah. So this will the title of my talk, from transcriptomics to proteomics, and the importance of integrated omics approaches in venom characterization. So I work as an assistant professor here, and we do have a mass spec facility which was funded by Agilent Technologies. So what I'm going to do is will give an overview about the work that I'm doing in my lab and the group which we are focusing, and sort of introducing the work. Okay, so it may be contain this work. I'm primarily focusing on giving an introduction about the characterization of the snake venom, especially focusing on the Western Ghats. So why snake bites or snake venom characterization? Because this gives an overall idea. This is a neglected tropical disease. And when you look into the figures, you could see that approximately 5.4 million people are bitten by snake bites every year, and resulting into almost 1.38 deaths, and 4 lakhs of them are permanently disabled. So it is very interesting to note that it's very interesting to note that the burden of death and disability is greater than any other neglected disease and equal to that of prostate or cervical cancer. So people always talk about cancers, diabetes, etc. So when we just figure out, or when we just compare with the burden, so we could see that it is equal to that of prostate or cervical cancer, which are predominant cancers in men and women. And this is a paper cutting from the Hindu paper. So, and this has been a report. Okay, which says that over 64,000 people die every year in India due to snake bites. Just because of these reasons, majority of the, a lot of snake bite burdens like morbidities and mortalities, World Health Organization has included the snake bite envenoming under the neglected tropical disease category. So these are some of the strategies that they have put forward to reduce the snake bite deaths, which includes improving treatment options, addressing antivenom issues, preventing and controlling snake bite, etc., etc. So when you look at, so this is the map that shows the distribution of the medically important snake species. So medically important snake species means they are a threat to the people or for whom these treatment options are not available. So when you look into the map, so this is the world map that says that there are almost 3,000 snake species, out of which 250 are medically important. So you look at here in the graph that shows the map of India, it says there are more than 18 snake species which are medically important. So these are the different biogeographical zones, Western Ghats, semi-arid desert, etc. All of you know that. So we are majorly focusing on the snakes which belong to the Western Ghats, and these are the big four snake species that are responsible for the majority of the snake bites death that is caused by the that is being projected earlier, that 1,38,000 deaths. So India houses, out of these 3,000 snake species worldwide, India houses almost 300 snake species, and among these 60 plus are said to be medically important. So two of them belonging to Elapidae class, and two of them represent the viper class. And these viper class of snakes or families, they affects the blood coagulation machinery, and the Elapidae family, they affects the or induce neurotoxic symptoms. Now, when a snake bites you, the treatment option, as all of you know, the antivenom is only the treatment option available, and it is being produced in the horses, and the usual procedure of making the antibodies. So basically, it's a decoction of the antibodies. So what is the real problem? What the real problem is, like when you look into the map, we could see that there are at least 31 snake species, medically important snake species, which has been reported, and this is already available in the WHO database. So now, when you look into this map, you could see that for majority of the snake bites, except these 4 snakes, majority of them doesn't have any antivenom available. Now, if you want to generate an antivenom, you should be having an idea what should be the complexity of the snake venom proteome. So, as projected by WHO, so this is the guidelines that has been provided by WHO. So, if you want to generate an antivenom, or if you want to control the snake bite deaths, you need to have an overall idea about the number of medically important snake species in a particular geographical region, characterizes the venom proteome, and provide the data or deposit the data in public repositories, publish manuscripts, and etc. And you need to perform WHO approved clinical trials also. So now we were majorly focusing on the venom characterization. So as I was discussing, one of the major problem that we face is the limitation of the sequences. So when you just look into the sequences of snake venom proteins in any of the databases like Uniprot or NCBI, you could see that the sequence information, the protein sequence information is very limited. So we need to perform only sequence similarity based approaches. So what we used to do, suppose so this is the basic workflow that we used to follow. We'll collect the venom, we subject to different separation strategies and subject to digestion, and later on MS analysis and database searches. So this is the overall workflow that we follow. And what we used to do is, like, even though we collect the snake venom from cobra or crate or Russell's or whatever it is, we don't have a specific database for the individual species. So what we used to do is, like, we'll select the serpentis, means it contains almost all the snake species and all the other reptilian organism that is falling under the serpentis. So we used to search, because as was mentioning, we used to perform a sequence similarity search because of the limitations in the database, sequences in the databases. And I'll just give an overview of what are the different snakes that we have characterized. So firstly, we have characterized the venom from Malabar pit viper. It's a pit viper which is found only in the Western Ghats of India, not to any other parts of the world, I would say. So we have identified 79 different proteins. So these proteins were identified by searching against the Serpentis database. And this shows the profile of the crude venom. Then we have characterized another species, which is also a viper. So this is also unique to Western Ghats. In addition to that, it is seen in Sri Lanka also. So the major problem with this snake species is often overlooked with any other name, because you might be misinterpreting this as a non-venomous snakes. But recently, the WHO or the medical clinical reports suggest that the venom from this snake species is very lethal. It causes majorly kidney injury. So what we did was, we collected the venom again from the Western Ghats. So we belong to Kerala, so we collected the snake species from Kerala, and we have characterized the proteome, and we could see that a total of 30 proteins. So you just look at the numbers. In the first species, there were only 79 proteins. In the second, there were only 30 proteins. So we were expecting more than 100 proteins in the venom. So, but somehow we were getting very less number. Then we moved on to characterizing the third species, that is the cobra, which is almost distributed throughout the Indian subcontinent, like Pakistan, Bangladesh, India, Sri Lanka, Nepal, etc. So even though this is widely distributed, what the interesting fact is, like, we have what we have done is, we have collected the venom and we have done the proteome profiling. And it's very interesting, because since this venom, because this snake species is distributed throughout the Indian subcontinent, there are a lot of reports. So when you look into the literature, we could see that at least there are 7 or 8 reports which will say the number of proteins identified and number of protein families. So all these proteins are classified depending on their clinical outcomes or the pharmacological actions. They are classified into different venom families also. So it's very interesting to see that even though it is the same snake species, you could see the number of proteins and the families identified is getting varied. So what does this say? Is it really the problem with the venom as such, or any limitation with the database? That's what we were interested on. I mean, interested in looking onto. And but however, keeping that point inside, we'll come to that later. We could see that from our study, from the venom collected from Western Ghats, we could see that a comparatively higher number of proteins and protein families, like 115 proteins. So how there is a variation, even though the snake species remains the same. Obviously, due to geographical variation, there will be some variation, but how drastic this change can happen within the venom proteome. So we just try to analyze, and we could see that even though there are at least 7 or 8 reports, we could identify 2 different protein families, like glutathione peroxidase and endonucleases, for the first time from the Indian cobra venom. And so that was the characterization part what we have done. And we were looking on to checking the efficacy of the antivenom. And since the geographical variation influences the antivenom efficacy, we were curious to know what is the efficacy of the currently available antivenom. So these are the 7 or 8 different anti-venom manufacturers in the country. So they used to procure venom from tribal people in Tamil Nadu, basically Irula society. So they used to collect snake venom and milk the snakes and give it to these different manufacturers. So they will manufacture anti-venom for clinical purposes and research purpose also. So interestingly, what we could see was we have initially compared the endpoint titration ELISA using different viper venoms and even Indian cobra venom. So what's interesting to note is like the efficacy is getting varied. Even though the venom is pulled from Irula and is manufactured in the anti-venom different 7 or 8 and anti-venom manufacturing companies, we could see that instead of having same efficacy, we could see there is a drastic variation. So what could be the reason? Is it real geographical venom variation that is attributing to the differences in the cross-reactivity profiles, or is it due to the problem with anti-venom manufacturing? We were interested to look upon, and these are the immunoblot analysis also. So this also clearly shows that there is some variation across different species and also within the snake species. So all these anti-venom, I mean ELISA or Western blotting, will give a qualitative estimation of the. Just a second. Qualitative estimation of the proteins, the efficacies. It may not be able to say what is the protein which is not getting neutralized or how the efficacy is getting reduced. None of these studies, because all these are qualitative analysis, and hence we have followed an anti-venomics approach. Basically, this you will be using an immunoaffinity chromatography matrix, which will be used to tag the antibody and will provide a venom onto that, and we'll see what portion of the venom is getting bound and what portion of venom is not getting unbound to the antivenom. So interestingly, we could see that these proteins, those which are highlighted here, these class of proteins are not getting neutralized by the antivenom. So what could be the reason? There could be multiple reasons due to geographical variation or due to the problem with the antivenom also. So that we need to dive deep into detail. Now, we moved a little further also. We have just taken this thing species from different geographical location and see how the intra-species venom variation, or how far the intra-species venom variation is there. So this is the Russell's viper venom. Okay, so this we have collected the venom from Tamil Nadu and the Western Ghats in Kerala. So from the eastern side and from the western side. So when you look into the STS phase profile, you could see that clearly there is a variation within the same snake species, and the numbers projects like there are the number of proteins and the number of protein families also getting varied. And since WRSLE or the Russell's viper is, again, since it is distributed throughout the Indian subcontinent, there are a lot of reports. However, we could see that those highlighted in the red color, like kallikrein, DNase, carboxypeptidase, a lot of cyclohexylene finger proteins. So all these proteins were novel and were reported for the first time from our studies. And since the proteins identified or the number of proteins are the quantity, when we look into the gel itself, we could see that just by looking into the band intensity, some protein families or the classes that is falling under the low molecular mass or high molecular mass range, they are showing the different band intensities. Obviously, when we load the same quantity of the venom, if they are showing a difference in the band intensities, this might give a clue like there might be the difference in the quantity also. Hence we thought of checking how far. Okay, so for example, we have taken phospholipases, L-amino acid oxidases. So these are the major venom proteins present. So we have done few functional assays to prove, okay, the real variation. So we could see that interestingly within the same snake species, you can see that the orange and the blue color. So we could see that there is a variation in the phospholipase activity, phospholipase A2 activity, and even L-amino acid oxidase. So this is one of the major protein that is causing for the lethal complications, L-amino acid oxidase, like majorly cell death and tissue disruption. So here also you could see that the blue and the orange, so there is a drastic difference in the protein activities also. And we have checked for an fibrogenolytic activity also. So fibrogenolytic activity means when a snake bites you, it might be interfering with a lot of body mechanism, and one among them is the blood clotting. So this is a viper venom, and we were interested to see how far this venom is affecting the or interfering the stability of the fibrinogen. And interestingly, we could see that. So this is fibrinogen with alpha, beta, and gamma side chains, and we could see that the venom from Tamil Nadu. We used to call it as RBVI, that is Russell's viper venom from Irula, Tamil Nadu. You could see that the alpha band is there at the worst hour, and it start decreasing as the time proceed, and parallelly the beta bands also, whereas the gamma bands, instead of getting reduced, they are getting increased. And similarly, in the Western Ghats, the Russell's viper from the Western Ghats, you could see that compared to the venom on the top, you could see that the alpha bands are completely disappearing even within 1 hour. So this was very interesting. What could be the reason behind this, and why this much of variation within the same snake species? And obviously, we have done the ELISA also, like we did for the other snake species, and we could see a drastic differences in the variation. Now, since, as I was mentioning, since the database information from the database regarding protein information of these snake species are limited, we thought of doing homology driven proteomics approaches using some de novo sequencing softwares. So this is the workflow that we have followed. We have done the collected the venom, separated the proteins subject to digestion. We created a peptide map, and if the protein is present in the database, yes, we have classified under different protein families. If not, what we have done is, like, we have done de novo sequencing using Novo, MS Blast, and even Peak softwares. So what does this say? Is, like, when the database has a limitation, we have done the de novo sequencing. We could see some protein sequences just like this. So this is a pictorial representation how we have done that. So this sequence was not there in the UniProt, I mean not UniProt, in the mascot or the common databases that are available. So but since this sequence information is already there, and we have performed a de novo sequencing analysis, and we could see that there is a change in the amino acid, means there is a mutation seen, that is the lysine in this 12th position has been replaced by arginine. So this is how we have complemented the conventional workflow with that of the de novo workflow, and clearly says that 5 to 15% of the peptides were identified through de novo approaches when you compare the conventional. So this says that additional or complementary sequencing analysis can also help to improve the confidence of protein identification. Now, as the title of my talk says, why from transcriptomics to proteomics, why transcriptomics? So, as I was mentioning, when you just type in snake species, for example, this is the UniProt result that shows the number of proteins in saw-scaled viper, and this is with respect to the Russell's viper. So when you see here, you could see there are only 16 and 33. So these are the total number of proteins that is already deposited. But when we do the mascot analysis, as I was showing earlier in cobra venom, we could see there are 115 proteins. How come this is there? So this is basically based on the sequence similarity, and might be representing, or might the sequences might be shared across different snake species. But to have an absolute quantification, for example, if you want to analyze the Indian cobra venom, or if you want to analyze the Russell's viper venom, you should be having an either the genome or either the transcriptome information of the individual organism as such. So this I would like to highlight the work which has been published in the Nature Genetics by a group. So what they have done is, like, they have sequenced the genome and the transcriptome of the Indian cobra venom, and they try to correlate with the transcriptomics data with that of the proteomics data. This will give an one-on-one comparison. If the protein is present, they will be saying, okay, this is there. So they could identify only 19 venom specific toxins instead of the 33, 40, 50, or 100, what the numbers which I was mentioning. So this clearly says that all the other database analysis, they were majorly focusing on sequence similarity, and they can't say, oh, this sequence is unique to the protein, or this peptide sequence is unique to a particular family. And since we have already collected the Indian cobra venom data, we thought of checking how far the different database analysis could give a difference in the number of proteins. So we have taken the Indian cobra venom from Irula, both Irula and the Western Ghats, and done different database analysis. For example, Serpentis, then Elapidae, Naja. So this Indian cobra, this belongs to the Naja species and Elapidae family, and we have further narrowed down into even to the predicted proteome. So the gland transcriptome data was already available, and we have just correlated the data with the transcriptomics data. We could see that, interestingly, you could see the numbers: 33 proteins, 35 proteins, 31 proteins, and 9 proteins. So the number is getting reduced. So this says that when we have the transcriptome data available, this will give a more accurate information. Similarly, with that of the Western Ghats species also. So this is the work that we majorly focus on, characterizing the venom proteome, and we are trying to correlate if at all there is a transcriptome data available, and we are planning to collaborate on sequencing the transcriptome if not available. So they to conclude to generate species specific antivenom, or to produce different types of antivenoms, unique to different geographical locations. You talk about anything related to venom, we need to have an integrated omics approaches. So otherwise we won't be able to specifically say this when this protein is unique to this species, or in the antivenom efficacy also. That's the reason why when you look into from an Indian perspective, or pan-Indian perspective, you could see that the antivenom that is available is polyvalent in nature. Means you have a decoction of the antibodies which is generated towards the all the four snake species, and not for an individual snake species. That is, no monovalent antivenoms are not available. So if you have these kind of approaches included, we will be having an more idea about the venom proteome. We can use those information to generate monovalent antivenoms also. And to further extent, we are working on developing some diagnostic kits also. So so that is the future perspectives that we are focusing on. So yeah, thank you. So I should acknowledge all these people, the funding agencies, ICMR, DHR, and Agilent Technologies, and the Forest Department people for helping with the collection of venom and helping in the analysis, and my wonderful collaborators and colleagues. And thank you, Dr. Prash, for this invitation. So as I was mentioning, this is the my blog. So I had shown shown only little bioinformatics. Behind the in the background, we are doing a lot of bioinformatics. So we are not bioinformaticians as such, but we just depends on the softwares. So as you have seen, we were depending on the different softwares for the collection of the data and analysis of data. And due to database limitations, we were facing a lot of difficulties initially. But then I used to have a conversation with Dr. Prash also on how to incorporate the transcriptome data to proteome data. And also, yes, I'll be happy to collaborate because as I was mentioning, analysis part was bit challenging for us for an unsequenced organism. Yeah. Thank you so much. So I'll be happy to accept a couple of questions.
Thanks a lot, Professor Muralidhar, giving us a very rigorous and extensive talk on the importance of integrating omic approaches to snake venom characterization. The bioinformatics aspects of this particular topic is very important, and it would be very nice if the audience here can collaborate with Dr. Muralidhar to produce more useful results. Currently, I would request the audience to ask questions if they have any. We will take about one or two questions. Yeah.
Yeah. Thank you so much, Murali sir. There are two questions. Barka and Kiran. Yeah, Barka, please. Yeah.
Hello, sir. Sir,
Yeah.
myself Barkha. It was a very intriguing talk. So great to
Okay,
listen to
ma'am.
you.
Yeah.
Just not a question, just one more kind of a suggestion. Like in our area, like in North and Rajasthan, there is a
Okay.
very learned professor, Professor K.K. Sharma. He's working on snake bite mitigation and everything. So, do
Okay.
you have any? We can take samples from the north as well, and if we may check the if there is a variation in the proteome as you have observed earlier.
Exactly,
That
that'd be
would
very
be,
nice because yeah. So as I was mentioning, since we are seeing a geographical variation. For example,
yes.
you catch a snake from the northern end and you compare it with the southern, and you could see a drastic difference in the composition. That could be due to a lot of reasons: geographical variation, then the diet, the prey which they are fed upon, even the sex also has an whether and the age, everything matters. So we don't know what is really contributing. And I want to highlight this: the government of India. So
Hmm.
they are keen, especially the ICMR. They are keen on giving funds to characterize the venom protein, especially to
Yes, yes,
northeastern
yes.
states.
Yeah,
The
because
problem
there
is
is
like
a lot of snake bites and snake, they're collecting it very much. So there are
yeah,
too many sightings even in the common areas, in the
yeah.
northern areas. So we should definitely connect with someone, and so that it would be a very nice study
Sure.
to
Yes, yes,
collaborate
yes.
on.
The major problem is like getting the venom. So, because since the venom is used for a lot of illegal purposes, the government has now, I mean, this year beginning onwards, has strictly imposed on using the venom or collecting the venom. So, we need to have, we need to follow rigorous ethical committee clearances, a lot of meetings and approvals from both the state and the central government institute, I mean, governments for getting the venom. So if you want to get venom, I think people from, for example, Northeastern should help us. If they are supplying the venom, yes, it's a direct or straightforward one. We can follow the conventional workflow, whatever we were discussing.
And just a small query, or as a layman, like here we used to mostly listen that the snakes that have been collected or they have been catched, they usually don't want to kill them, and they want to they are considered to be non-venomous. So how exactly is the ratio of the snakes found in the southern area? Like these are mostly venomous or non-venomous that you
So
usually found?
yeah, so I can just give an clarity from on with regard to this from the Western Ghats perspective. So, basically focusing on Kerala, I would say there are approximately 210 snake species so far reported. So, out of this, it's interesting to note that 97 percentage, or I would say 98 percentage of them are non-venomous. Only 2 percentage. So, I would that the list I have shown. Like the 37 snake species that is being reported from India, they belong to the medically important. And when we talk about the venomous snake species, people only know about these 4 snakes. So the cobra, the crate, and the 2 vipers. They rest all the other snakes. Even if you even if you are bitten by a non-venomous snake, due to the I would, what should I say? They are afraid of snakes, first of all. So whenever a non-venomous snake bites, also they are keen in catching the snake and take it to the hospital. So so don't do that. So that's a
Because
kind of
mostly people are so scared of it, they consider all the snakes to be dangerous. And
exactly.
instead
So
of
that is not the scenario.
catching
That
it
is
or doing it, mostly people go for killing them and getting rid
yeah
of them. Yeah.
yeah.
Yeah, Dr. Murka, you can ask more questions during the bus stop. Yeah. Uh,
Okay, thank you, sir.
Dr.
Thank
Kiran,
you, thank
please
you.
ask your question. Yeah.
Okay. Yeah, thanks. My question is very short. I just wanted to know whatever you're trying to do, like as you said, the collecting the information is a bit restricted and everything is that. All the information what you're collecting is publicly available. If so, what is the resource? If you can provide that, that would be great.
Yeah, so what all information, what are the data that we have generated from the mass spec is already deposited in the Pride and Protein Exchange. So you can get all this information. So far we have deposited the 3 information about the protein information about 3 snake species, 2 vipers and 1 elapid. It's already available in the Pride repository. I can give you the accession number so that you can download it, and if you want to analyze it, you can try reanalyzing that also. So that's the only way of depositing these information, protein information.
Thanks a lot.
Okay, thank you.
Yeah, thanks a lot, Mr. Muralidhar, for giving us a very interesting and a talk which is directly applicable to end users and
Yeah.
quite applicable to very applicable to our country. And thank you very much. And then with
Welcome.
this, we'll end this session. Yeah.
Sure. Thank you so much. Thank you all.
Thank you. Yeah, thank you. Thank you so much, Murli, and thank you so much, Jaraman, sir. Just a point on Barca. Barka, Professor K.K. Sharma, they're already a part of that Unapoya University's, anti-venom project actually. Murli must be knowing. So they were all, forming a consortium.
Okay.
Which, of course, Amrita University, they were also invited at some point of time. Murli, Barka is a postdoctoral scientist. She'll be very happy to connect you to, K.K. Sharma. Yeah, please. Huh? Yeah, thank you. Thank you so much, Murli, for your illustrious talk. Thank you so much. Yeah.
Welcome.
Yeah.
Thank you.
So, yeah. So next we have.